their cameras to preserve bandwidth, and so that the speaker can be heard with clarity. Please follow these guidelines. Also, do not share your screen at any time during the talk, because this completely messes up the system. And when the speaker is presenting, pin the presentation to your screen so as not to lose. And today we're going to make the session a bit more interactive than usual. So the speaker might ask questions during his talk and expect answers from you, the audience. Understandably, it's a bit more difficult to regulate this virtually. So please bear with us and wait your turn. I would urge people to use the chat box. So what you can do is, when there is a question, you can post either your name or something saying, I want to answer. And I will pick out a name, and then I will call that name out. You can unmute yourself and answer and discuss and engage in conversation. But when you're done, also please mute yourself so that the presentation can continue smoothly. I also again repeat, please maintain decorum, and people who don't may have to run the risk of being removed from the session, unfortunately. Let's hope that this does not have to be the case. Of course, as in every other week, you can pose questions to the speaker, and this you can post to the chat box, which will be taken up for discussion at the end. Another thing, the feedback link will also be given to you towards the end of the session. I'd now like to hand over the session to our convener, Professor Inare Banerjee, to welcome the speaker of the day, Professor Sourav Ghosh, to our webinar series and open today's session. Over to you, ma'am.
Thank you, Shreya. The excellent Krishna Shahu had started presenting. So this is again something that we should be a little conscious about. And if there is a presentation, then Sourav's screen. Sourav, our speaker, and a very warm welcome to all the participants tonight. I think we have a YouTube platform which is being also the archive biology was conceived, is being managed and maintained entirely by a group of postdocs and PhD students. So, and from around the world. So this is a platform that is for facilitating a meeting of the minds, and we are very fortunate that we've had some of the sharpest scientific minds of our country, of our times, of our country and abroad, happily, to come on our platform and share their scientific work with the participants, which have undergraduate students, postgraduate students, and doctoral scholars and postdoctoral fellows, and very young to very, very senior professionals inside and out of science. So the platform is to engage, to network, to collaborate, to get ideas. And I think Sourav today, Professor Sourav Ghosh from Yale University, who is happily our alumnus from the Department of Zoology, which has just completed its 100 years. He is for a somewhat different way of engaging with the audience today, and actually, for the next 1 week, we are having the final exams of all the graduate and the postgraduate batches of BA, BSc, BCom. So we see a bit of a dip in the participation tonight. But whoever's here, this is a rare opportunity to engage with our speaker tonight. So a very warm welcome, and I request Shreya to please introduce our speaker. And then, as we say in Bangla, Sourav, the Moidan is all yours.
Thank you. Thank you, Anna.
Thank you, ma'am. So Professor Sourav Ghosh did his BSc and MSc in zoology from the University of Calcutta in 1990 and 92, respectively. He then finished his PhD in molecular sciences from the University of Tennessee in 2001. Professor Ghosh served as a postdoctoral fellow at the Salk Institute of Biological Sciences till 2007. He then joined the University of Arizona, served as an assistant professor till 2011 and an associate professor till 2014. Professor Ghosh has been an assistant professor at Yale University School of Medicine from 2014 to 17 and an associate professor at Yale from 2017 to 19. He's currently an associate professor with tenure at the Department of Neurology at the Yale University School of Medicine. He's also a member of the Signal Transduction Group at the Yale Comprehensive Cancer Center since 2014. He has several awards and grants to his name and has published over 30 research articles in peer-reviewed journals and 18 chapters, books, and reviews. Professor Sourav Ghosh is here today to speak to us about hysteresis in T-cell activation. A very, very warm welcome to you, sir, to our platform, Progress and Prospects in Biology, and the stage is all yours, as ma'am said.
Thank you. Thank you so much, Shreya. It's really an honor to present in this forum. Let me just go to see if you can see my presentation. I am presenting, should be. All right.
Yes,
so,
we can see.
Can everybody see my screen? Okay. So thank you again. And I'm really happy, especially to see some of my classmates from zoology. And you know, this is truly a pleasure that such a beautiful series have been organized by PhD student postdocs with the guidance from Anna. Anna, thank you so much for putting this together. This is really nice. I realized that it would be difficult for me to try to do a conversational style, so probably I'll try to stick more conventional. And if there are questions, Shreya, if you don't mind unmuting yourself and asking questions, referring the questions, because I'm not really looking at the screen. I'm only looking at my presentation.
Yeah.
So anyway, let's get started. What I'm going to talk about today is some work being done in our lab at Yale, and it's to do with T-cell activation. And it's really something that was unexpected. We are calling, the title of this presentation will be Hysteresis in T-cell activation, and I'll explain, go along. But I don't know sort of the background of the audience. I presume it's a fairly broad audience from very different disciplines. I myself was a student of zoology. We used to call ourselves Bang, like a frog, So from how do a frog, how can somebody who dissects frogs know so much about immunology? So I thought I will start with a little bit of an introduction to get everybody on the same page, and then we'll go from there. We'll try to explain the data. And if anybody wants, cannot follow, if they have a question, please feel free to interrupt. Please stop me. Okay. So what are T cells? I mean, I think we are all familiar with T cells. These are very important cells in the adaptive arm of the immune response. So when, let's say you have an injury, the first cells to respond would be the innate immune cells, such as macrophages, dendritic cells, and then eventually the problem with innate immunity, it's very fast, but it's very broad. It's non-specific. It doesn't know who it's attacking, right? So there are no real good targeting mechanism. But eventually the immune system sort of transfers over to its second arm, the adaptive immunity, the T cell and B cell mediated immunity, where the target is very specific. So whichever pathogen is bothering you, the immune system is now going to go after it. So these T cells, and in particular is the subject of today's presentations, they're derived from progenitors in the bone marrow, from hematopoietic stem cells. And these cells, they come to the thymus, and they're selected as T cells, right? CD4 positive, CD8 positive, so there's a lot of selection about that happens in the thymus. So that's where the term T cell is from thymus. And then these naive T cells, they enter the circulation and essentially go through your bloodstreams, right? So it passes from your bloodstream, enter a secondary lymphoid organ, a lymph node or the spleen, and then they leave and go back to the bloodstream and migrate, so they get into the tissue through the blood, they get out of the tissue through the lymph, and this circulation continues till in the secondary lymphoid organ, they encounter dendritic cells presenting their cognate antigen, and that's when the T cells become activated. So this is sort of textbook knowledge. So what happens at the lymph nodes is a dendritic cell will present, hopefully you can see my cursor, will present the peptide on its MHC that is recognized by the TCR in the T cell, and there are some other signals like CD8086, CD28, and especially for CD4s, there are other signals involved as well called co-stimulation, and this is what is going to lead to activation of this T cell. And this is textbook. So Charlie Janeway was an immunologist at Yale University, and we follow his textbook. I think his textbook is quite popular in the US. So this is from the 9th edition of the Janeway Immunology, and you can see the way so this is a chapter on T cells, T cell activation, and what is in the textbook is that, yes, it's the MHC presenting the peptide to the TCR that drives T cell activation. And then there are co-stim signals, which in its absence, if you get the first signal, T cells will become actually anergic. So these are sort of survival signals, and then there are cytokines for differentiation. But everybody agrees that naive T cells are activated just by the TCRs getting stimulated. So naive T cells are generally considered, they're quiescent cells, they're just simply going around the body. They don't have much diversity in them. This is a homogeneous population, except of course they're TCRs. So T cells, they are clonal in the sense that they have very different TCRs, and each TCR can see a specific peptide when presented on MHC. And when the T cell gets activated, they proliferate, and then you get clonal expansion, right? So this is sort of our baseline. Everybody with me so far? That the naive T cells are considered a homogeneous population, okay? And once T cells are activated, there is a lot of diversity that people have already recognized that happens, and these are examples in CD4s, the helper T cells. So you have Th1, Th2, Th9, Th17, Th22, TFH, Treg, many different types of T cells, right? You don't have to know the details. These are the different cytokines that help the T cells differentiate. So they have each expressed different transcription factor, different gene expression signatures within them. And that's why they're called these different subtypes, right? But all this was thought to happen only after a naive T cell gets activated, all right? Now, this concept is beginning to change. So this year in Science, there's a paper from El Tamboulis, who's the first author, where if you see where my cursor is, what they did was do something called single-cell RNA-seq in T cells from the spleen, naive T cells from the spleen. And what you can appreciate, so each dot here, let me explain the data, it's one single cell. And what this dot represents is the entire transcriptional profile, the transcriptome of a single T cell. And so if you do this kind of experiment where you take a bunch of T cell and you learn the transcriptional profile of each individual T cell, then what you can do based on similarity between two cells, you can make this sort of clusters. So all the red cells, they're all unique cells, but they have similarity in their transcriptional profile. Once they did this study, they realized that you have these red clusters and green. Of course, the colors are fake, they're superimposed. It's just the transcriptional diversity. So, you can group this naive T cell, the so far believed to be the homogeneous population of T cells, turns out to actually not be so homogeneous, but be consisting of different subpopulations, okay? So that you can have a green sort of group of naive T cells, blue, red, of course, these are pseudo colors. You can imagine like an analogy would be like, all the people listening to me, if I ask you to stand up and then I organize you based on your heights, right? So the taller people, above 6 feet, let's say it's the red group, then 6 to 5'5" or whatever below. So something like that. So this is not height, this is the entire transcriptional profile. But what this tells you is that not every T cell is the same, the naive T cell. So this is sort of the background we need to follow the story that I'm going to tell you today. And the work was done by Alex Melly. He did his PhD in Canada in McGill and came to the lab as a postdoc. And Dimitri, he was an MD PhD student in the lab. He finished his PhD and is now doing his clinical work, getting ready to do his fellowship. And I co-lead my lab along with Carla Rotlin. Many of you might have heard her presentation in this forum. All the work that we do in collaboration. And one more thing about the story today, there is a lot of bioinformatics because we did a lot of this single cell RNA-seq, bulk RNA-seq, different techniques. So there's a lot of bioinformatics, and that was mainly done by a postdoc in the lab, Yachu Wang, but he was guided in all the bioinformatics by Molloy Kumar Basu. And the reason I mention this is Molloy, who is now at UAB in Alabama. You can see him here. We were classmates in zoology, the 91 batch, the masters in zoology at Calcutta University. So this is Molloy Basu. He's an assistant professor at University of Alabama, Birmingham, and we collaborated. And he basically guided Yachu for all the bioinformatics. So it was quite nice, to collaborate with somebody from my school days or my college days. And the work that I'm going to present today, it's all entirely unpublished. Hopefully it's publishable. It has been deposited to something called bioRxiv. Now a lot of people, the preprints of their papers, when it's ready to be sent out for review and publication, potential publication, we upload our work to bioRxiv so that it's available to everybody across the world. You can access and find out the recent research that's going on, because typically publication, from the point now I'm presenting this work, probably it will only be published a year, 2 years later. I would be lucky if it is published, accepted in a year, because there's usually a lot of comments that you have to address and things like that. All right. How are we doing in terms of time? I think I'm good. All right. So what Alex did first was isolated naive T cells from the mesenteric lymph nodes in mice. And he did the single cell RNA-seq, just like the El-Tambuli work that I indicated. And indeed, we can replicate the discoveries by El-Tambuli. So we also see different heterogeneous subpopulation of naive T cells when we isolate the naive T cells from mesenteric lymph nodes. And these are all naive T cells. We are very careful in terms of all the markers. I'm not going to go into the immunological nitty-gritty and details. That would be boring. But take my word, and you can look up in the paper all the markers we used to isolate the naive T cells by flow. And then we subject them to the single cell RNA-seq. So we can replicate the work. What's the big deal? The big deal is the question that we decided to then think about is, what is the significance of this heterogeneity in naive CD4 T cells? They're already diverse in terms of their TCR, and as far as we understand, all you need for activation is to tickle or stimulate the TCR. So why do we need this transcriptional heterogeneity in naive CD4 T cells? What does it do? Is there any functional importance to it? So to try and address this question, what Alex and Dimitri they did was use a model of Helminth infection in mice. So we use this nematode, Nipostrongylus brasiliensis, which infects mice. And once it's infected, it's a very well-established model. It's a sub-Q skin infection. And then the larva gets to the lung. And then from the lung, the mouse kind of coughs it up and swallows it. So the Helminth now gets to the gut. And that's where the adult helminths reside, and then they're expelled, right? So in a Th2 immune response dependent manner. So a lot of things happen in the gut of these mice, and that's why we decided to focus on the mesenteric lymph nodes. And what Alex did is compare the heterogeneity in naive T cells in non-infected versus in Nipo-infected mice. And when he did that experiment, we were really, really excited and amazed, because I'm sure you can realize what the difference between a non-infected and Nipo, correct? See, so I highlighted this. You see this population, which is marked, which is the green population, cluster 2, that there were very, very few cells of that type in non-infected mice. But upon infection by Helminth, in the Helminth that goes to the gut, and we are looking at the mesenteric lymph nodes, you suddenly see this population come up. So this changes, this heterogeneity in naive T cell, it's not a fixed heterogeneity. It is a dynamic change that happens when the mice gets infected. So this was really an exciting finding. And again, this is just a control. We are very sure that the change that is happening is not reflective of activation of naive T cell. These cells are still naive. This is the single cell data looking for different markers of naive versus Th2. And you can see the activated cells, which you will get activated T cells, CD4s, and which will differentiate once the mice have been infected with Nipo. And you can see they cluster very, very differently from the naive cells. So the conclusion so far is that naive T cell subpopulations are not fixed, but change dynamically in response to infection. And this is an absolutely new discovery. We are super excited by this. This was not known. But of course, you don't believe in just one observation to say that this is real. So we, of course, wanted to know what is the mechanism of Helminth-induced changes in heterogeneity of naive T cells. What is the mechanism? Because if we find the mechanism, we can trust our results more, right? We feel better about it. So the first clue came when we tried to look at expression of the top 20 genes that marked cluster 2, right? So this clustering is done based on the gene expression profile. So we just took the top 20 genes that are highly expressed in cluster 2 and is lower in the other clusters. What stands out to us is that these genes, actually many of them are known to be transcribed when cells see IL-4. This is IL-4 receptor. Even the IL-4 receptor that can be upregulated, you see that's the third one from the top, that's part of this 20 gene signature for this cluster 2. So that suggests that it might have something to do with IL-4 signaling. And these are genes that are top 20 genes that are downregulated, and a lot of them are the IFI genes. So it's the interferon responsive genes that are going down as the IL-4 genes that are coming up. So we thought, yes, maybe let's think whether IL-4 is the mechanism to make this new naive T cell cluster. And so we looked at expression of some of the genes, some of the components of IL-4 signaling machinery. So that the IL-2 gamma common chain, the IL-4 receptor alpha, those are indeed present in the naive T cells. Other Th2 cytokines such as IL-5, IL-13, components of those signaling, those receptors were not present in the naive T cells. So we started to think this is probably IL-4 driven. And so next what we did was now we took the naive T cells and did bulk RNA-seq. Okay, so now we didn't do single cell level RNA-seq. We lysed all the cells together instead of individually, and so we could now look at the bulk RNA-seq. And the bulk RNA-seq, if you look here of Nipo over non-infected, many of these genes that were upregulated were also IL-4 dependent. And that became very obvious because not only we did the experiment comparing Nipo-infected with uninfected by bulk RNA-seq, what we did was inject the mouse with an IL-4 complex. So this is IL-4 bound to an antibody to stabilize it. You inject it in mice, so the mice is getting this IL-4, and compared the genes that are regulated by IL-4 over non-infected. And then what we did was look at these two data sets and the overlap. And as you can see, significant fraction of the genes that are upregulated by Nipo infection are also upregulated by IL-4 infection. Of course, IL-4 induces more genes, but our interest was this set of 84 genes, so there is a lot of overlap. So we thought, okay, this sounds like Nipo infection, which is known to drive IL-4, is probably what is causing this, this subpopulation level difference. So Alex did one more experiment where he took non-infected mouse, Nipo-infected mouse, injected with an isotype, a control antibody, and Nipo-infected mouse, but now with an antibody that will neutralize IL-4. So it's an anti-IL-4 antibody. So now the antibody will bind IL-4 and stop it from binding its receptor and signaling. And then did bulk RNA-seq in these three groups. And what you can see is the genes that are turned on upon Nipo infection, the ones that are shown in red over here, you can see there's many genes. When Alex neutralized IL-4, those genes were no longer induced, unlike with Nipo alone. So that clearly, mechanistically shows that Nipo infection and the consequent IL-4 is what's driving this T-cell, naive T-cell heterogeneity. So that's, again, we concluded that changes in naive T-cell subpopulations is brought about by IL-4, a cytokine associated with Th2 response during Helminth infection. So what are the functional implications of these changes in naive T-cells? And to test that, what we did was infected mice with the Helminth, with Nipo, and then we vaccinated the mouse to study its immune response. We used OVA peptide to vaccinate along with alum as the adjuvant. So this is a type 2 adjuvant. It's used in many vaccines. And the reason we actually did this experiment, I'm going to come to it at the very end, it is known that in countries where there is endemic Helminth infection, kids often do not respond very well to vaccination. Okay, so we wanted to see if the Helminth infection and these changes in the naive T-cell subpopulation, does it functionally affect response to vaccination? So to do that, Alex, like you can see, if he looks at the draining lymph nodes in these mice, there's a lot of cells. So the lymph nodes are expanding. That indicates that there is a response going on. If you compare that with the lymph cells in the cellularity of the lymph node, from Nipo-infected mice, there is a significant decrease. This is just a control, non-draining, nothing is happening there. And when he looks at CD4, CD19 B cells, again, there is less once the mouse has been infected with Nipo. When you vaccinate the mouse, which is carrying a Nipo infection, they are not responding as well as a non-infected mouse. Alex also looked at IL-4 cytokines, such as Th2 cytokines, such as IL-4, IL-5, IL-13, and all of these are depressed in the Nipo-infected mice. He looked just as a control to type 1 cytokine like interferon gamma. There's obviously no effect because those are not supposed to go up in an ovalum infection. So, this is an important control because it suggests that the response is not switching from a Th2 to Th1. It's still a Th2 type of response, but depressed. It's a weaker response compared to uninfected mouse. And here he measured antibodies, and again, also at the level of antibodies, the anti-OVA antibody, the mouse doesn't make very much anti-OVA antibody. So their whole response to vaccination, or which could be a secondary infection also, would probably do the same thing. We get a weaker response. Of course, we are not content with just looking with one Helminth because there could be some artifacts. So Alex also used H. poli. This is another Helminth infection in the mouse and gets essentially the same results. In the interest of time, I'm not going to go over with all the details, but again, we see less T cells, less Th2 cytokines, less OVA-specific antibody. And he also wanted to change the adjuvant. We were not sure whether this effect is only specific to alum, Th2 type of response. So he switched the adjuvant to a Th1, Th17 adjuvant CpG, and in there also the response seems to be lower. So now, of course, you're getting this interferon gamma, IL-17, and those were now dampened in the mouse which is infected. So what can we conclude? We conclude that T cell subpopulations are not fixed, naive T cell subpopulations. First of all, they are heterogeneous, and this heterogeneity changes dynamically in response to infection. This is brought about during Helminth infection through IL-4, and that functionally is associated with dampened response in the mouse to vaccination, right? So everybody with me so far? Okay, so now what we needed to prove, so far we have an association, right? That there is a change in heterogeneity of the naive T cell happening, which is correlating with the dampened response. But can we show causality? Can we show that the changes are indeed because now the naive T cells are different? So the way we set about doing that, we first ruled out other cells that can lead to this difference. So for example, you can imagine in Helminth infected mice, the DCs are not presenting very well. They're not migrating to the lymph node. So therefore the T cells are not getting activated. So we first, ruled that out by looking at DC specific ovas that's draining, or looking at T independent responses, right? So there are certain immune responses which are driven by which are T cell independent IgM type of responses, and there were no differences in these mice. So that's sort of a control. So then what Alex did is he infected mice with Nipo, a CD45.2, or it's a slightly different mouse than it's congenic, so there's not going to be rejection, but we can find the T cells if we transfer the T cells from this mouse to this recipient mouse. One is 45.2, one is 45.1 positive. So what he did was take the T cells from an infected 45.2 mouse, put those T cells in a 45.1 mouse, then challenge this mouse with vaccination, okay? And then he analyzed what the response is happening. Because now you can trace the T cells that were given to this mouse that came from a Nipo-infected mouse. And what he found that those T cells did not proliferate as much as the T cells if you transfer the T cells from a non-infected mouse to a non-infected mouse. So this sort of indicates that the T cells coming from a mouse that have been infected are not very good at responding, right? They're not proliferating. But you can still argue that, because this is also an infected mouse, there are some changes that we don't understand yet that is causing that. So then Alex did the really, really key experiment, which is take T cells from a mouse that was infected with Nipo, transfer it to another mouse, but this time he's transferring to non-infected mouse. Okay, so these are not infected at all, right? Unlike the last experiment. And even here, what he found was that these T cells coming from an infected mouse, they were proliferating less than the scenario where non-infected were transferred to infected. So this clearly suggests that the T cells themselves are carrying some sort of a defect in activation when they're coming from a Nipo-infected mouse. So indeed, the failure of the mouse to produce an immune response can now be ascribed to these naive T cells that he's transferring from an infected cell. So we looked at gene expression in these T cells, and we know at a gene expression level as well that the T cell function, this categories like chemokine genes, T cell function associated genes, cell cycle associated genes, these are dampened in the T cells coming from the infected mouse. So this was also an interesting finding, more molecular correlate of the proliferation. And finally, because I have shown you before that we think all these naive T cell changes are happening because of IL-4, Alex wanted to test if now this infected mouse, if we give anti-IL-4, which will block the IL-4 signaling, and therefore this heterogeneity is not going to happen, the change in heterogeneity, which I showed you at a transcriptional level. If we take T cells from this mouse and transfer it to a non-infected mouse, we shouldn't have this dampening anymore, right? We should get the dampening still in the Nipo-infected, but we can prevent that by anti-IL-4 treatment. And indeed, that's what he gets. So here again is T-cell proliferation. This is CFSC dilution assay. And you can see that the Nipo-infected, the T-cells are not proliferating very much. But if we take naive T-cells from Nipo-infected mice, which also got anti-IL-4 blocking antibody, now the proliferation is more or less like wild type, right? So they're proliferating more than if isolated from the Nipo-infected mouse. So now we can say for sure that it's the naive T cells that are carrying some sort of an activation deficit, which of course is an IL-4 dependent effect on these naive T cells. So we felt, this really convincingly proves that for the first time that naive T cell subpopulations are not fixed but changes in response to infection. It's driven by IL-4, it's associated with reduced response, and indeed changes in naive T cells can be directly associated or ascribed with this reduced vaccine response. So therefore, we decided to call this hysteresis in T cells. So what is hysteresis? Hysteresis is actually a non-linear phenomenon in mathematics, in electrical circuit design. What it simply means that the output is dependent on the initial state of the system. So the output is not just dependent on the input, but also on the initial state of the system. So let me give you an example of this in terms of circuit. So if you think that an input signal U, when it's less than S minus R, the response, the output will always be -1. If U is greater than S plus R, then of course the signal is going to be +1 over here. But in this range, in this type of a circuit, in between S minus R to S plus R, if U falls somewhere in between, whether the output would be +1 or -1 depends on whether the signal is increasing, right? So if the input signal is increasing from S minus R towards S plus R, as soon as it crosses S minus R, it will become +1. But if it is dropping from S plus R towards S minus R, it would remain, it will immediately go to -1 as soon as it crosses over from S plus R to this side. So in this range, the signal, whether it's +1, the output, whether it's +1 or -1, depends on what was the state of the system. Was it here before or was it here before? Okay? And this is illustrated by agar, for example. I don't know many of you, whether you know this or not, but you know agar, right? And agar solidifies somewhere between 32 degree to 40 degree, right? But it melts at 85 degree. So between 40 to 85, its present phase depends on whether it was previously a solid or a liquid. So let's assume you take solid agar at 40 degree. It has never been melted, right? And it will remain a solid when you heat it to 60. It will only melt when it's 85, right? So at 60, it's still a solid. But now if you take melted agar at 85, and then you start cooling it, at 60, it will already become, it will be a liquid when cooled to 60 degree, right? So it's not going to become a solid till the temperature drops farther. So at 60 degree, it can either be a solid or can be a liquid, depending on whether you started off with solid agar or with liquid agar. So this is called hysteresis. So the initial state where you found it is super important. So it's not just the input that you give, but the state of the system was existing in together gives you the output. This is also known, it's very important for drugs. So for example, if you, I take blood pressure medication and I forgot to take it yesterday night, let's assume, the concentration is not going to immediately fall because there is a hysteresis, right? The state of the system has been changed. So even if you take the drug out, it's still maintaining itself. So there are examples in pharmacodynamics of hysteresis as well. So it's super important phenomenon in electricity, in biology, in chemistry. I was joking that if this paper gets rejected, calling it hysteresis, I'm going to resubmit it by saying T-cell activation is regulated by karma. As an Indian, maybe people would accept that, coming from an Indian. Because this is like whatever your past karma was decides, depending on what you do today, taken along with your karma, your fate is decided, right? Not just on your actions today. That's sort of our Indian concept, and that would be an equivalent to what is hysteresis, okay? It's kind of a joke, but it's roughly equivalent. All right? So hysteresis can be two different types of hysteresis. It can be rate dependent, it can be rate independent as well. So rate independent is a persistent memory of the state. It's a permanent effect. The rate dependent is where the initial input, like the Helminth infection, if it's cured, then the effect will go away, right? Then the T cell, this heterogeneity will go away. If it's a rate independent effect, the heterogeneity would stay. So we decided to check that. And what we did again is single cell RNA-seq. But this time we did it at 1 week after Nipo infection, which is the peak infection period. And Nipo is naturally expelled by the host. So by 3 weeks, there is really Nipo infection has been cleared. So Alex did single cell RNA-seq in non-infected, 1 week after infection and 3 weeks after infection. And I hope you can appreciate that at this cluster 2, it was non-existent in non-infected. It was very large and significant at week 1. But by 3 weeks, as the infection has been cleared, this cluster is going away on its own. Okay, so this is a classic rate-dependent hysteresis event, where the infection is gone, the T cells, the state of the cells returns to what it was before, and then you will end up with a system, hopefully like the non-infected very soon. And this is just more bioinformatics way of indicating that indeed that is the case. I'm going to skip it in the, it's not super important. We also did the same thing by bulk RNA-seq. So we did bulk RNA-seq at 1 week and at 3 weeks. And again, you can see all these genes, Nipo-dependent genes that were upregulated at 1 week in these naive T cells, they are going away. So again, it's the same thing. We like to show it with different independent techniques. Furthermore, what we did was, okay, we checked now the effect on vaccination, which I showed you, this dampening effect, was it also going away as the Nipo infection is cleared? So now what Alex did was waited for 14 days after infection, a further 7 days, and then gave the ovalum vaccination. Because generally by 14 days, the infection is gone. So we just wanted to make sure it's absolutely gone. So we wait 3 weeks. And then he vaccinated and then looked at the vaccine response. And here you can see the vaccine response was identical 3 weeks later in a Nipo-infected animal. So it's the persistent infection is required for this dampened vaccine response. And okay, and this is just the IL-4 response, like the same responses that I was showing you to you earlier, which was dampened in Nipo-infected mouse. Now, if you wait to vaccinate up till the infection has been cleared, there is really no difference between non-infected and Nipo-infected animals. This is antibody response. Oh, we also used a separate model of H. poli. H. poli now is a chronic infection, unlike Nipo. So it's not cured by itself, but you can cure it with a drug. So what we did was, again, give the mouse the H. poli infection and then treated it, gave it some rest, and then vaccinated. And now, again, the dampening effect of the Helminth infection with H. poli, which I showed to you before, is also gone. So this shows that this hysteresis effect on the T cells, the reshaping its initial conditions, it requires a persistent infection, so it's a reversible event. Okay, so I've been talking for a while, 15 minutes to go, 45 minutes I've been talking. What's the implication of all these discoveries that I want to share with you? So first, this is really a novel, fundamental understanding of T cell activation, that the initial state of the naive T cell really, really matters. So before this, all the textbooks will tell you that T cell activation only requires MHC peptide TCR interaction and co-stim. But what I'm trying to tell you today, if I'm correct, is that these signals are not just the signals that drive T cell activation, we have to take in consideration the initial state of the T cell, whether it was like this, or like this. All these changes in T cell, the naive T cell transcriptional pool, which is dynamic change, happens based on the history of the organism, of the individual, whether they have been infected or not, does indeed impact on how easily the T cells can be activated. And what I showed you in Helminth, in the particular case of Helminth infection, the response threshold, the magnitude of response is dampened. So let me give another example. So if I ask you, how do you boil water? You'll say, okay, I have to heat it. Sure, you heat the water undergoes a phase change and becomes vapor, right? Now, heat is like the equivalent of, in the context of a T cell DC interaction, it's the equivalent of this MHC, CD80, CD86 interaction, right? So this is the external energy that is being provided into a system, right? Into the T cells by the DCs in this case. But what is also important for boiling water, and the analogy here would be, if I now, instead of giving you fresh water, if I take water from the Dead Sea. So the last time I love to travel, and the last time I could travel before this COVID thing was, we were in Israel in January, and that was the last trip. And there's, the Dead Sea, the Salt Sea in Israel. And there's a lot of salt in the water. So if you take water from the Dead Sea and if you heat it, you can imagine it's not going to behave just like fresh water. The boiling point will be higher. So the initial state of the system really does matter. It's not just the heat. And similarly, in the context of T cell, it's not just the signals being provided by the APCs. It's the initial state of the T cells are important. And not only is this sort of a conceptual understanding, this finding also provides an understanding of epidemiological data. So as I mentioned, that it is known that parts of the world where there is endemic Helminth diseases, the vaccination response is really, really poor. So there is a strong geographical overlap with regions targeted by vaccine campaigns, and the response in these places are usually poorer. So there are multiple papers saying human infection with Ascaris, for example, is associated with problems with vaccination against cholera. There's many BCG. There are different ones, and poor immunogenicity of BCG in Helminth infected population, right? So it's the same as the mouse if it's infected with Nipah, or we think the mouse model we are using is a model of diseases in patients, which is a new mechanism to explain why these kids do not respond very well to vaccination. So in the context of concurrent infection with Helminth and another disease, the immune response would be weaker, or in the context of vaccination, it wouldn't be very efficient. But there is a flip side to this, and that would be the hygiene hypothesis. So hygiene hypothesis says that with reduced disease burden in advanced and developed countries, the burden of atopic diseases like atopic allergic diseases or like chronic inflammatory diseases such as IBD, inflammatory bowel disease, is on the rise. And that, the idea is if we had Helminth infections, maybe we would not be so prone to hyper-responsive autoimmune, chronic inflammatory diseases. And for this, I will point out to some bioinformatics that Molloy actually did for us. So he looked at the single cell data, and he tried to predict what are the origins of the clusters, what are the trajectory of origin of these clusters. And he found out that all these different, the cluster 2, 3, and 4, the one characterized by IL-4, one characterized by interferon, and the TCR sort of cluster, we don't know, what drives that. They all come from 0, 1. Cluster 0, 1 are the source clusters. So this is sort of a trajectory. The cells come like this, and then they bifurcate, and they have a choice at this point of either going this way, or this way. So we found that this cluster 4 and cluster 1, they are sort of a specific bifurcating choices. So you can either go on the direction of IL-4 cluster or to type 1 interferon cluster. And we think that this is what keeps the immune system sort of in balance, these competing clusters, IL-4 cluster and type 1 cluster. So that helps set the magnitude. And since we sort of co-evolved with Helminth, in the presence of Helminth, you have these two clusters that sets your baseline response. Okay, it might be harmful in the context of vaccination, but it's beneficial in the context of avoiding autoimmune chronic inflammatory kind of problems, such as IBD. But now, in the modern world, with sanitization in the developed world, you do not have Helminths anymore. So now all the naive T cells are poised or biased towards this interferon cluster, which now correlates with a higher magnitude of response in individuals. So they're at a hair trigger response, right? And so that's why maybe a lot of these chronic inflammatory diseases are in the rise in the developed world. So now that's pretty much all I have to say, and I will in a moment try to answer some of your questions. But let me acknowledge, my professors, Ashish Dattagupta and Probir Chakraborty, because as I said, I was a zoology student. I had no idea about immunology. And it's only Probir taking classes that was organized by AKDG after hours when we were zoology students that I got my alphabets in immunology. And I cannot imagine that today I will be talking to you about some advances in immunology that we're really, really excited about. We think this would be materials for future textbooks. Anyway, the program, as I said, is directed by Carla and I. We, of course, acknowledge the funding, without which the work would not have been possible. Single-cell RNA-seq is quite expensive. And of course, all the lab members. We have many different projects in the lab. We have a lot of interest in cell death. We are doing some neuroscience, studying glial responses in neurodegenerative diseases. We have focus on cancer. We have quite a diverse program. You can look at our websites. And what I would do is at the very last, I would prop this other series of talks called Global Immunotalks that Carla hosts along with Elina Zuniga. And these are immunology talks and accessible all over the world. It's free. It's also posted on YouTube. And they really have an amazing list of speakers, all like really the top immunologists. Internationally, they have, this is just the second series. There was some earlier. And I think from what I understand, they're going to continue in 2021 because of the interest that they have heard. So if you have time, check this out. Okay, and thank you very much. That's all really what I have to say. I'm going to stop sharing screen and see if I can answer questions. I think we did finish within time. Thank you.
Yes, I think so. Thank you, Professor Ghosh, for a delightful talk. It was very well explained and very easy to understand, I would say. And there's a lot about T-cell activation that at least I didn't know that I learned. So we have a lot of questions, both on this platform and from YouTube. And I'll try to ask the most pertinent ones, and forgive me if we run out of time on some others. So if we can begin. So the first question says that why is it that only mature dendritic cells activate naive T cells? Is there any mechanism?
So this is basic immunology. You can find it in textbooks. So it's immature DCs are really, really good at phagocytosis. So they can take in dead cells and process it. So they are also good at migration. But once they phagocytose, they start maturing. And then they can migrate. So they can no longer phagocytose, they migrate to the lymph nodes, and then they present the antigens. So this is how it is. This is how it is done. We have not only the mature starts expressing the MHC peptide on the surface and activate. Yeah, when they're immature, they're busy eating, sampling the antigens, and then when they mature, they present it. Okay. I think I saw one question. I don't know if Shreya, if you don't mind, I'm breaking the rules perhaps, but somebody asked, is this true for naive B cells? And we think so. So we are going to collaborate with Eric Meffrey. He's a professor here. He's an expert in B cells. And, in theory, this should apply. This cytokines are diffusible through it's found in circulation. You can measure it in circulation. So all cells would be exposed. And certainly B cells, I think this is a fundamental principle. It should apply to B cells, perhaps even macrophages in terms of their polarization. I think this is the beginning. This is the tip of the iceberg. We're super excited. And it's not just Helminth, we are now trying to work. There are many different experiments we are trying to do with trying to see what affects this diversity and what this means. Okay, sorry, Shreya.
No, sure, absolutely. You go ahead. So another question is very relevant in today's time, of course. So what are the role of T cells in vaccination, and particularly in the case of COVID? Is it known?
You know what? I refuse to touch COVID. I understand the need for the study, but I'm not a virologist. Well, I guess I was not an immunologist, but we have been studying immunology for a little while. We've been trying to understand. But from what I understand, I can tell you what I have read in the literature. So, and this was, I tried to read about more the SARS and the MERS viruses, because they're the same family, it's not COVID-2, it's COVID-1. And there are reports that there's not much of a B cell response, right? So the response is T cell driven. And so that's it's not, you will generate memory T cells perhaps. I don't remember literature showing that, which will give you some protection, but it's not as great as B cell antibody. But there's no reason why, I don't know. Vaccines, if you can elicit a response or not, I guess that's where you're going. Whether the vaccines would work or not, I don't know. I really have not, I can't speak to that.
Yeah, it would be more speculative probably than anything. Okay, sure. So another question is there any interaction between the genes in cluster 3 and 2 that you showed? Because the loss of cluster 3 and the increase in 2 at 1 week post-infection gets reversed on day 3 post-infection. So your comments.
Very good observation. Super good observation. Yes. We don't know what gives rise to the interferon cluster. So we know that Alexander Rudensky at Sloan Kettering, his group, they have been working on, I believe, viral infections, which seems to, this is not published, this is yet. Well, I don't know. So other groups are studying this. I don't think we know yet the origin of, or things that affect the interferon cluster, and it would be very interesting. So I think it's going to be different infections that you get that might change the distribution between the different clusters. Some through interferon, type 1 interferon signaling, the other through IL-4 type of signaling. And these should be in balance. And it would be super interesting. We are really interested in, looking at this, and other groups are looking at it as well. So it's, again, like we hit upon the Helminth model, the other groups working on different models, and the naive T cell heterogeneity, I think there's going to be a lot of growth in this area.
Mm-hmm. So another question relating to this cluster is that, have you found a mouse mutation which results in high numbers of this cluster 2? Does it exist?
High number, okay. I mean, you can say that if the IL-4 receptor is mutated, a loss of function, then you would lose this cluster, but that would be terrible. I don't think there is an IL-4
Could probably not survive.
So that's a really good question. There might be polymorphisms that make the naive T cells more responsive to IL-4. It's possible that there is a polymorphism that now, even at very low levels of IL-4, there's going to be a signal and you will generate this. This is a really, really good question. We have not looked at that. This is super new. It's not published yet. But yes, this is a really good question, and it would be interesting.
Yeah. So another question from Prabhat Kumar Swine. So how did synthetic gene networks help in hysteresis in your study?
Synthetic gene networks. Well, look, the reason I'm calling this hysteresis is that there was not an appreciation that the state of the naive T cell had anything to do with T cell activation. So it was all the input signal coming from MHC peptide through the TCR, right? And also the co-stim that people considered as the activating signal. And I actually chose to call this hysteresis because what I think the point that we are trying to get across is that it's not just that signal, but the initial state of the T cell, whether it was sort of preconditioned or pre-primed by IL-4 or by interferon, type 1 interferon, or some other diffusable cytokine. Because so when you get a Helminth infection, you will be responding against Helminth-specific antigens, right? So the Helminth-specific naive T cells, which has TCRs towards those antigens, they would be getting activated in the secondary lymph node, and there's going to be a lot of IL-4 produced in that context. So the other naive T cells that are there will see the IL-4, and also IL-4 gets into the circulation, and so we haven't looked at other lymph nodes, but we are thinking about looking at the spleen and other places. It's not just going to be restricted to the mesenteric lymph node. Perhaps the IL-4 will get to the other lymph nodes, and then those cells will be preconditioned, and that initial state will reflect when they get their specific cognate peptide, next time around, like for example, when you vaccinate. So this concept is what hysteresis is, that the initial state of the system affects the output from a specific input, and that's why we call it. The subpopulations we were able to identify based on their entire transcriptome, that there are subtle, like some differences in the gene expressions. That's why they clustered like that.
Okay, thank you. If you have time, could we ask a few more questions?
Of course, yeah, I have time.
Okay. So there's another question. It says, expression of CD4 cells show a sigmoid expression. So is CD4 high cells and CD4 low cells and CD4 intermediate cells showing the same type of cytokine secretion pattern during the infection, or is it different?
So we haven't sorted based on CD4 expression to see if this
right.
is how it happens. So I don't really know that specific answer. I'm seeing Shubhro Chatterjee. He's also listening to me. Shubhro was also a classmate. No, so we don't know. And I don't think it depends on CD4. I think the CD8 cells, we have not looked at naive CD8s, but I think, again, they might have the same sort of effect. So this is just like cells that have the receptors for the cytokines. They will be exposed to the cytokine, and there is going to be what we are finding is they have some transcriptional changes happening within them that is changing this initial state. So I do not know specifically, but I would assume, it's not dependent on that.
So there's another question from Momita Shah. She says, does the transcriptional regulation of naive T cells have any effect on autoimmune diseases, and thereafter making the immune response of a person so weak that it gets a reduced response to vaccination?
Right. So this is known epidemiologically that kids with Helminth infection, they don't respond well to vaccines. You can deworm them, and then they respond better to vaccines.
Mm-hmm.
Regions which there is endemic infection, the kids keep getting Helminth infection. So that's why sometimes the vaccine response is not good. Now, there could be, I'm not claiming that this is the only reason for that. Look, there is at an organism level, even if you have a Helminth infection, your overall metabolic state probably is a little bit energy deprived. There's going to be effects on all sorts of aspects of the immune response. But it is interesting that this pre-priming, this hysteresis of these naive T cells might contribute towards that. That's all I'm trying to say.
Yeah.
The quantitatively, how bad is the effect? We don't know. We have just modeled it in mice with sort of an optimum Helminth infection, like sort of the standard protocol. So these are probably more epidemiological questions that I don't have the expertise to address. The other side of it, we do think that lack of Helminth or the lack of this IL-4. So we work with SPF, right? Specific pathogen-free mice.
Yeah.
It would be really interesting, and I'm giving away ideas here to do it in dirty mice. So we do think about collaborating with people who are working with pet shop mice and seeing what city, and those would be interesting things to pursue, and we are very interested in pursuing, in trying to do those. It is possible that having a larger this interferon, the IL-4 type of cluster correlates with reduced autoimmune or chronic inflammatory diseases. Potentially possible. We don't know the answer, but that's what would be my guess.
Right. So based on that, there is another question. So Piali Chatterjee asks, does this mean that we